Hello, I would like to present the poster Antibody Validation and Antigen Discovery by Combining Protein and Peptide Microarrays. Pepperprint is founded and based in Heidelberg. We're operative since 2011 as a privately held independent company with a proven track record. We're ISO 9001 certified in the quality management system and currently count 25 employees. The company has developed a solid-state printing technology that enables the generation of linear and cyclically constrained peptide libraries by successive and stepwise coupling of individual amino acids based on toner particles in situ, that is directly on the chip surface. Available microarray formats range from the discovery format, shown on the left, with up to 75,000 peptides, to the standard array formats in the middle, which can be further partitioned into smaller subarrays termed as mapping and multiplexed formats with as little as 273 peptides per individual array. Note that the peptide spots are by default displayed as duplicates. Here we present an approach to accurately determine antibody specificity by combining protein and peptide microarray screening of two different commercial research antibodies targeting human transglutaminase 2. In particular, we are examining a mouse monoclonal antibody by the company Zadira and a rabbit polyclonal antibody from Atlas Antibodies. We proceeded in three different experimental phases. First, a broad cross-reactivity profiling on the human protein microarray, then printing the obtained hits for linear and conformational epitope mapping validation on a pepper chip custom microarray, and lastly we carried out a substitution scan whereby every single amino acid in the epitope is stepwise replaced by all the other 19 amino acids and examined for overall binding strength. This allows to identify conserved and variable amino acid positions. Here we have the results from the human protein array screening of the mouse monoclonal antibody with the main response against TGM2, weaker cross reactions in red, and in blue, the rabbit polyclonal antibody with strong cross reactivity on the protein level and no detectable response against TGM2. The results from the multiplexed hit epitope mapping show a clear monoclonal response against the N-terminal epitope in TGM2 for the mouse monoclonal antibody and a strong polyclonal response against the C-terminus of TGM2, as well as multiple validated cross-reactions against other hit proteins. The outcome raises questions about the reliability of some validation methods, such as immunohistochemistry, Western blot, or RNA sequencing. The combined protein and peptide microarray screening, however, is not only an ideal tool for antibody validation, but also for the identification and validation of unknown antigens. The epitope substitution scan of the wild-type peptide shows 7 well-conserved amino acid positions. A high number of conserved amino acids generally reduce the probability of off-target binding. In summary, the three-step validation workflow: first, the protein hit discovery, followed by 2, multiplexed epitope mapping for hit validation, epitope identification, and 3, the epitope substitution scan for the analysis of conserved and variable amino acid positions shows that the rabbit polyclonal antibody strongly cross-reacts on the protein array, however less so on peptide microarrays, where it did show strong target specificity. This was also in accordance with the expectations. The mouse monoclonal antibody has a higher target specificity and less off-target binding, with an epitope of 7 well-conserved amino acid positions. This slide briefly summarizes the antibody validation workflow. Please contact us if you have any further questions.